Assalamu alaikum dear students, welcome to your channel The Anatomy
So
Canvas.
in today's class we are going to discuss with the histology of the bone, but before going on to the next slide, first subscribe to the channel, press the bell icon, and I highly appreciate your comments in the comment box. So learning objective for today's class are to discuss the functions of the bone, little bit about the functions, then the composition of the bone and its different constituents, different types of the bone, and to discuss the different types of the cells which are forming the bone. So we will discuss all of these objectives one by one. As a main constituent of the adult skeleton, the bone tissue provides solid support to the body. This is very important function and it protects the vital organs such as those present in cranial and thoracic cavities. Also encloses the internal medullary cavities, the present within the bone which contains the narrow spaces in which the blood cells are being formed. In addition to support and protection, the bone plays a very important secondary role in homeostatic regulation of the blood calcium level. This is very important function of the bone. It is in constant condition of homeostasis with the blood calcium levels under the effect of the two hormones secreted by the thyroid gland and the parathyroid gland. Bone is a specialized type of the connective tissue that is characterized by mineralized extracellular matrix. So this is the diagram of a long bone in which you can see this is the proximal epiphysis, then the area of the metaphysis, then the shaft or the diaphysis, then again the metaphysis and epiphysis. The epiphyseal area is covered by the articular cartilage, and this area you can see the spongic bone is present. And in the center you can see the medullary cavity which is lined by the compact bone. So these are the different types of the bone which are forming the structure of the long bone. So basically bone consists of the three main elements as all other connective tissues. We describe all of the connective tissue under the three headings: the types of the cells, the extracellular matrix and the connective tissue fiber. So in case of bone, these are bone cells, the extracellular matrix and the connective tissue. The very important feature that distinguishes bone from other connective tissue is mineralization of its matrix, which produces an extremely hard tissue that is capable of providing support and the protection. So this mineral component which is present in the form of calcium phosphate forms the hydroxyapatite crystals. Now the bone matrix, we will first discuss the bone matrix, and about 50% of dry weight of the bone matrix is inorganic material. The calcium hydroxyapatite is present, it is most abundant, but maybe the bicarbonate, the citrate, the magnesium, potassium, sodium are also found in it. But mainly it is the calcium hydroxyapatite crystal which is present in a form of the inorganic material. The significant quantities of non-crystalline calcium is also present, and the major organic structure. So these are the inorganic material. The organic structural components of the bone is mainly the type 1 collagen, and to lesser extent type 5 collagen, and type 3, type 9 and type 13 are also present in traces. But main organic component of the bone is the type 1 collagen. The all collagen molecules constitute about 90% of total weight of the bone matrix. So we see that 50% of the dry weight of the bone matrix is inorganic, and out of this 50%, 90% is formed by the collagen molecules, and remaining 10% will be formed by the non-collagenous molecules. So non-collagenous molecules constitute ground substance of the bone and 10% of the bone matrix. There are four main types of the non-collagenous proteins. This is also important. The proteoglycan macromolecules which are attached to the gags. These are the core proteins of the proteoglycan monomers. Then the multi-adhesive glycoproteins, these are also very important because because of these multi-adhesive the glycoproteins, the bone components like collagen fibers, the osteocytes, the osteoblasts, they are attached to the bone matrix. Then the bone specific vitamin K dependent proteins which include osteocalcine. This osteocalcine is very important because it helps to maintain the release of the calcium, absorption of the calcium, and maintain the homeostasis of the calcium. Then the growth factors and the cytokines which are also important. These cytokines are secreted by the osteoblast upon stimulation by the parathyroid gland, and then they activate this osteoclast for the resorption. So these cytokines and growth factors, they are also very important. So these four components, these are the non-collagenous proteins: the proteoglycan, the multi-adhesive glycoprotein, the bone-specific vitamin K dependent protein, that is osteocalcin, and growth factors and cytokines. These are the non-collagenous proteins. Then bones are organs of the skeletal system. We know all very well. Bone is the structural component. Bone tissue is the structural component of the bone. And then bone tissue is classified either as compact bone or as the dense bone. So in this diagram, you can see this is the epiphysis and the metaphysis of the long bone. This is the shaft or the diaphysis, then the metaphysis, and again the epiphysis. And here you can see the epiphyseal line, where before adult life, when bone is growing, there will be epiphyseal cartilage which will help to increase the length of the bone. Also in this area, in the epiphysis, you can see the spongy bone is present, while the lining underneath the periosteum and also the lining of the marrow cavity is also the compact bone which is present. So these are the two different locations of the bone. The spongy bone in the marrow cavities it is present, and the compact bone either beneath the periosteum or just next to the endosteum. Classification according to shape. Bones are classified according to shape, like location of the spongy and compact bone varies with the shape. So they may be the long bones that are longer in one direction, that is humerus and femur. The short bones nearly equal in length and diameter, having the compact and spongy bone with narrow spaces. Articular surfaces are covered with hyaline cartilages like carpal and the tarsal bone. Then we have the flat bones. These are the thin and the plate-like bone like the scapula and the sternum. And then we have the irregular bones, the vertebrae, the hip bones, and ethmoid. So basically, according to the shape, the bone are classified into four main categories: long bones, the short bones, the flat bones, and the irregular bones. So general structure of the bone, this is also very important. Bones are covered by the periosteum. So outer covering of the bone is periosteum, except at the articular surfaces where a dense fibrous connective tissue, where the articular cartilage is present. So this periosteum is a sheath of dense fibrous connective tissue containing the osteoprogenitor cell. Here you can see in this diagram that this periosteum consists of the two layers. The one is the outer layer, which is a fibrous layer, and next is the inner layer, which is more cellular layer. It contains the osteoprogenitor cells. Then the collagen fibers of the periosteum are arranged parallel to the surface of the bone in the form of the capsule. So here you can see these are the collagen fibers, their arrangement in periosteum. Very important, another structure, that is collagen fiber. From these structure extends directly but an angle into the bone tissue called the sharpest fiber. So these sharpest fibers are the type of the fiber where the tendons of the muscles are attached to the bone, so give strength to the tendons of the muscle as well as the bone. Here the collagen fibers are present at an angle, where we are called these fibers as sharpest fibers or the perforating fiber. Next, the structure of the bone, you can see this is the osteon of or the eversion system of the bone. This is the basic structural component of the bone, the structural organization of the bone. So in this eversion system, in the center you can see the eversion or the osteonal canal is present, and in this is one of the osteon. You can see in the cross section, here you can see in the osteonal or the eversion canal, you can see the blood vessels and the nerves and the lymphatics are present. Around this osteonal canal, you can see the collagen fibers are arranged in the form of the lamellae. And here in this section you can also see this is the arrangement of the collagen fibers. In a given lamellae, the collagen fibers are parallel to each other, but they are at right angle to the collagen fibers in the next. So here the angle between these these two lamellae between the collagen fiber is 90. Also in between these two lamellae you can also see one is in this direction and other collagen fibers are in this direction. So this is a very important arrangement of the collagen fiber to give the strength to the bone. So this is the one of the osteon, in which you can see in the center you can see the osteonal canal or the Haversian canal is present, and around this Haversian canal the lamellis are present. The collagen fibers are arranged in form of the lamellis, and in these lamellis there are the spaces you can see in this enlarged view. In these spaces are called the lacunae. In these lacunae you can see the osteocytes are present, and these osteocytes have the dendritic processes called the which are present in the canaliculi. So through these dendritic processes, these osteocytes have the gap junction with the neighboring osteocytes, and with this they can contact the other osteocytes. They have both the gap junctions with the neighboring osteocytes of the same lamellis as well as the adjacent lamellis. So by means of the gap junction, the osteocytes are attached to each other, and they transmit information to each other. So this is one of the osteon. This is detailed structure. In center you can see osteonal canal with blood vessels, and then the lamellis containing the lacunae, the canaliculi, and in lacunae you can see the osteocytes are present. In the canaliculi you can see the osteocytic processes are present. So in this diagram also you can see this is one of the osteon, this is another osteon, this is third osteon, and these are the different osteon. So in between these osteon you can see the interstitial lamellis are also present, and outer to these osteon just beneath the periosteum you can see the external or outer circumferential lamellis which are present. Similarly, just closest to the endosteum you can see the inner circumferential lamellis are present. So these lamellis are arranged in different types. One are forming the osteon or the Haversian system, other are present in the form of the interstitial lamellis, another are present in the form of the outer circumferential and the inner circumferential lamellis. So the blood vessels which are present in the osteonal canal, different osteons are connecting to each other via the perforating canals or the Walkman canals. And in through these Walkman canals, different osteons have connection with each other. So bony cavities, the marrow cavity and the spaces in the spongy bones contain the bone marrow. So these are the different marrow cavities. In you can see the blood vessels which are also present in the marrow cavity. The red bone marrow normally restricted to spaces of the spongy bone in adults, and the yellow marrow consists mostly of the fat cell. It can revert to red marrow, for example, in cases of the extreme blood loss. So this is the capability of the yellow marrow to revert to the red marrow when there is the extreme blood loss and body needs the blood cells, so these cells changes to the red marrow. Now we will discuss the endosteum. Here you can see this is the lining, inner lining which lines the inner side of the bone cavities is called the endosteum. This is a layer of the connective tissue and it contains the osteoprogenitor cells. So lining tissue of the both the compact bone facing the marrow cavity as well as the trabeculae of the spongy bone within the cavity is referred to as endosteum. So here you can see this area from here to here, this area is the spongy bone. I can mark it for you. From here to here you can see this is the spongy bone. So I change the color when I'm so from here to here you can see this is the compact bone, and this area is the spongy bone. So you can differentiate between the two. This area is the compact bone in which you can see proper arrangement of the osteonal system, the lamellis all is present, and also these are the irregular bony spicules of the spongy bone, and surfaces of both the spongy bone and the inner surfaces of the compact bone is lined by the endosteum. So this endosteum is lining of these areas in it contains the osteoprogenitor cells. Endosteum is often only one cell layer thick and consists of osteoprogenitor cells, and that can differentiate into bone matrix secreting cells, osteoblast and bone lining cells. So these are the different types of cells which can differentiate from the osteoprogenitor cells. So we can again discuss the bone as the mature bone and the immature bone. So mature bone, as I have told you, is composed of the structural units called the osteons or the Haversian system. The osteon consists of the concentric lamellae. These are the concentric lamellae of the osteon. They surrounding the central canal, and the central canal is also called the osteonal or the Haversian canal. It contains the blood vessels and the nerves. Here you can see this is the osteonal canal. This is again the osteonal canal. The canaliculi are generally arranged in a radial pattern with respect to the canal. So in the canal, this is the osteonal canal, and these are the arrangement of the canaliculi, which contains the lacunae and canaliculi which are present over here, and their arrangement, you can notice their arrangement. Then I have told you the interstitial lamellae which are present in between the osteons. Then I have told you the circumferential lamellae, the outer circumferential which is just beneath the periosteum, and the inner circumferential lamellae which is just closer to the endosteum. Then we have the perforating canals or the workman canals, through these workmans or perforating canals, the two osteonal systems are connected to each other. So this is the ground section of the bone. Very important point to notice over here, that in a ground section we can very well see the architectural details of the bone, but it is difficult to see the cellular details of the bone. So for the cellular details we can see the H and E stain section, and for the architectural details we can see the ground section. So this is the ground section, this is the low power of the ground section, and this is the higher magnification of the ground section, in which you can see this is one of the osteon. In the center you can see the Haversian canal is present, and then you can see the circumferential lamellae of the osteon or the Haversian system. In these circumferential lamellae you can see different lacunae which are present, and radiating from these lacunae you can see the canaliculi. This is the enlarged, further higher magnification of this section. So you can see in these areas this is the lacunae. In the living bone it contains the osteocyte, and from these lacunae you can see these are the radiating canaliculi which contains the processes of the osteocytes. So here also you can see these are the different Haversian systems which are present. This one is the interstitial lamellae which is present. This one is the Walkman's canal, and this is the circumferential lamellae, maybe the outer or the inner depending upon the location where it is present. So first generation osteon in the developing bone, you can see how the osteons are forming. Then when osteons mature and next generations osteons are present, the previous osteon forms the interstitial one. So the interstitial lamellae are formed from the the first generation osteons, and then the second generation and the third generation. So this process keeps on, and the bones mature itself. This is again another section of the compact bone. Here you can see one of the osteon. This is the another osteon, and in the center of the osteon you can see the osteonal or Haversian canal is present. These are the circumferential lamellae of the osteon or the Haversian system, and these are the interstitial lamellae which are present. Here you can see these are the perforating canals or the Volkmann's canals which are present, and you can differentiate these Volkmann's canal because osteonal canals runs parallel to the long axis of the bone, while perforating canals are present at transverse axis. So you can differentiate between the two very well. Now we move on to the spongy bone, and this is spongy bone, the mature spongy bone, is similar in structure to the mature compact bone, except that the tissue is arranged as trabeculae or the spicules. So these you can see irregular bony spicules or irregular bony trabeculae which are present enclosing the marrow cavities. These are the marrow cavities, and these are irregular bony spicules of the spongy bone. In this also the bone is arranged in the form of the osteons as well. So numerous interconnecting narrow spaces of various sizes are present between bone tissue, and matrix of the bone is also lamellated. Just like compact bone, this is also lamellated bone. It contains the osteons as well. So next we move on to the immature bone. So far we were studying the mature bone. Next we move on to the immature bone. The skeleton of the developing fetus, also called it forms skeleton of developing fetus. It is also called the bundle bone or the woven bone, and because of interlacing arrangement of the collagen fibers. Comparison, we can compare it with the long bone, mature bone, that it does not display an organized lamellated appearance. In this you can see large number of the cells which are present, and osteocytes and osteoblasts both are present. In the center you can see the osteonal canal, but still you cannot appreciate the proper lamellated appearance of the bone. The more ground substance is present in this bone, and it is less heavily mineralized. That is why it stains intensely with hematoxylin. The area of the immature bone are present in it, especially at the site of the remodeling. So this is the comparison of the immature bone and the mature bone. In mature bone you can see this is the H and E stain section of the mature bone, in which you can see this is the osteonal or eversion canal, and this is the complete osteon, in which you can see circumferential lamellae, and here you can see the osteocytes which are present in their lacunae. Here you can see this is the immature bone. These are the bone lining cells which are forming the bone, and the irregular bone irregularly arranged fibers are present, and this there is the less eosinophilia and more staining with hematoxylin because it is less mineralized. Again, you can differentiate in diagrammatic presentation. This is the immature bone and this one is this mature bone. Large number of cellularity, decreased number of the cells, the proper development of lamellae, that is the osteons. There is no proper arrangement, and it is not less mineralized, it is more mineralized. So now we move on to the cells of the bone tissue. There are 5 different types of the cells which are present. First of all, the osteoprogenitor cell will differentiate from the mesenchymal cell. These osteoprogenitor cells then give rise to 3 types of the cell: the osteoblast, the osteocyte, and the bone lining cells. The fifth type of the cell which are present in the bone, also called the osteoclast, are derived from the granulocyte monocyte progenitor system. So bone tissue cells are surrounded by the matrix. They contain 25% water, 25% protein, and 50% mineral salts. So first of all, we will study the osteoprogenitor cell. Here you can see osteoprogenitor cells which are present. Osteoprogenitor cell is a resting cell that can differentiate into an osteoblast, and it secretes the bone matrix. They may be the periosteal cell when they are lining the periosteum, called the periosteal cells. When they are lining the endosteum, they are called the endosteum cells. These cells are basically flattened or squamous shaped with lightly staining. They have elongated or ovoid nucleus and inconspicuous acidophilic or slightly basophilic cytoplasm. Electron microscopy reveals profiles of RER and free ribosomes, as well as small Golgi apparatus and the organelles. So these are the features. On light microscope, you can see slight eosinophilia or slight basophilia, the flattened shaped cells with the ovoid nucleus. And on electron microscopy, you can see the profiles of the RER, the Golgi apparatus, and the free ribosomes. Next are the osteoblasts. This osteoblast is a versatile secretory cell. It can secrete both the type 1 collagen and the bone matrix proteins. So these bone matrix proteins include the calcium binding protein such as osteocalcine and osteonectin. Also contain multi-adhesive glycoprotein, that is the sialoprotein 1 and 2, the osteopontin, the thrombospondin, various proteoglycans and their aggregates, and also secrete the alkaline phosphatase. This is also very important. So in this tissue you can see these are the cells. These are cuboidal shaped cells. These are the osteoblasts. These are present in the area where bone formation is taking place, and these cells, this is the spatial stain in this slide. These cells secrete osteoids. This osteoid contain the organic component, but it is less mineralized. That is why it is stained deeply intensely with the basophilic dyes. So on light microscopy, you can see these are the cuboidal shaped cells which are forming, which are the osteoblast, and their aggregation into single layer in a position to forming bone. So this is the section of the bone where the osteoblast are present in a single cuboidal shaped cell. Also, in addition, you can see over here the osteoclast is present. When these osteoblasts lay down the bone, they are and the bone matrix, which is the osteoid. This osteoid is present. When this osteoid is mineralized, these osteoblasts are trapped and then they become the osteocyte. So this is the schematic diagram. Here you can see the osteoblasts are present, then the newly formed osteoid which is present, and this is the bone which is mineralized, in which osteoblasts are present in the lacunae forming the osteocyte. These are the marrow spaces, and this is the osteoclast which is present. So cytoplasm of the osteoblast is markedly basophilic. This is a very important point again. The Golgi apparatus, it contains Golgi apparatus, and because of its size is sometimes observed as clear area adjacent to the nucleus. So on electron microscopy you can see that these are basophilic cells with a clear area around the nucleus. They have the PAS positive granules in cytoplasm, and a strong alkaline phosphatase reaction associated with the cell membrane can be detected by appropriate histochemical staining. So in this slide you can see this is the area, this is basically the area where the extracellular matrix is present, and this extracellular matrix contain osteoprogenitor cells, the blood vessels, the nerves, lymphatic, and the osteoblast as well. So these osteoblasts develop from the osteoprogenitor cell. They are cuboidal shaped cell and they secrete the osteoid. This osteoid is non-mineralized, so you can see and you can differentiate. This is strongly stained with the basophilic diet. Then you can see when mineralization takes place, this osteoid is mineralized, the entrapped osteoblast now become the osteocytes, and here you can see the osteoclast which is present, and this osteoclast is present in a lacunae which is called the house shaped lacunae, where are the resorption bay which is present over here. This one is the electron micrographic picture in which you can see different osteoblasts which are present, and they have secreted the osteoid which is present. These are the small canaliculi which contain the processes of the osteocyte, and here you can see these are the this is the matrix present, extracellular matrix. Here you can see the entrapped osteoblast now become the osteocyte. So this is electron micrographic picture of the osteoblast. You can differentiate it. Osteocyte is the mature bone cell enclosed by bone matrix that is previously secreted as an osteoblast. So as I have told you, these osteoblasts, and they differentiate into the osteocytes. These are the cells responsible for maintaining the post bone matrix. Large number of the cells, main population of the cells are the osteocytes. Normally most abundant in the bone, they contain significant osteocyte exhibit significantly less rough endoplasmic reticulum, small Golgi complexes, and more condensed nuclear chromatin than the osteoblast. Because these three features, the RER, the Golgi complex, and chromatin, they are present in the osteoblast, which are the properties of the protein secreting cell. So as these cells are responsible for the bone formation, so they have the large profiles of the rough endoplasmic reticulum, the RER, the Golgi apparatus. But in contrast to these osteoblasts, these osteocytes contain significantly lesser amount of rough endoplasmic reticulum, the Golgi apparatus, and the ribosomes. So again, the three types of the osteocytes again are present. These osteocytes are the quiescent osteocyte, contain fewer profiles of the RER, and this is the osteoid lamellas which are present. Here you can see this is the formative osteocyte. This osteocyte resembles a little bit the osteoblast. It contains larger profiles of the rough endoplasmic reticulums, and here you can see the arrows shows the presence of the collagen fiber fiber. So these are formative osteocytes somewhat resemble osteoblasts because in condition of stress they have the capability to form the bone matrix. Similarly, another type of the cells, the osteocytes, are the resorptive osteocytes. They also contain the rough endoplasmic reticulum, but important point is that they secrete the metalloproteinases, and these metalloproteinases digest the collagen fiber. So in close vicinity you can see the clear area which is present over here, and this shows that the osteocytic area of osteocytic osteolysis. So they also contain the lysosomes as well. Again, this is the electron microscopic picture of the osteocyte. This osteocyte is present in its lacunae, sending the small processes. These are the cytoplasmic processes of the osteocyte, which are present in the canaliculi. So this is the diagram again showing the osteocytes and their processes which are present in the canaliculi. Again, you can appreciate in this ground section as well. Third, another type of the fourth type of the cells which are present are the bone lining cells. These are derived from osteoblasts and cover the bone. Some book categorize these cells as the fourth category, and some books describe these cells as a type of the osteoblast. So these osteoblasts, when they are quiescent, they are lining the periosteal or the endosteal sides, and they are called the bone lining cells. So here you can see these osteoblasts. There, here you can see these are the bone lining cells, and here you can see these are the bone lining cells which are present, and they are attached to each other by the gap junctions. Again, you can see these are the bone lining cells which are present attached to each other by the gap junction, and this is the external lamina, the plasma lamina of these bone lining cells. Last type of the cells which are present are the osteoclasts, and these osteoclasts are derived from the monocyte macrophage system. They are large multinucleated cells. Their size vary according to their activity. The active cells are very large. They may contain up to 50 nuclei in their cells. So when these cells are present, they are present in a bay called the resorption bay, or the house-shaped lacunae which is present, and also in this lacunae these cells have the irregular cytoplasmic processes which are called the ruffled border. In addition, so here at these ruffled border they have the ATP dependent proton pump, and these cells are related to the osteoclastic activity, that is resorption of the bone. In addition to this resorption bay, here the area is called the clear zone or the sealing zone, and this zone contains, in this area, the no subcellular organelles are present, and this is the compartment at site of the ruffled border where resorption and degradation of the matrix occur and resorption materials are absorbed. So this is the structure of the osteoclast. It contains large number of the lysosome, and this cell is activated by the cytokines which are released by the osteoblast. So these osteoblasts are activated by the hormones secreted by the parathyroid gland. These osteoblasts secrete cytokine, and these cytokines then activate the osteoclast. So remember, these osteoclasts do not have directly their receptors for the parathyroid hormones. So they are activated by the cytokine, and upon the activation they secrete the lysosome, and they causes the resorption of the bone. So that is the end of the 2 days class, a little bit lengthy class. These are the references: the basic histology by Laikus and Siddiq, the histology text and atlas by Janquera, and histology text and atlas by the Hraj. Thank you very much for your attention, and Allah Hafiz till the next class.